Today we will talk about the tryptophan operon. This is tryptophan operon. Just like the lac operon, this tryptophan operon, it explains the gene regulation in prokaryotes. This tryptophan operon, the concept of tryptophan operon, it was given by Francis Jacob and Monod. This tryptophan operon, it is a repressible operon. 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 What is meant by repressible operon? It means that when the extra tryptophan is added in the culture medium, no enzyme synthesis occurs. When we add the tryptophan in the culture medium, no synthesis of enzymes occurs in E. coli. So it is a repressible operon. That means extra tryptophan present in culture medium, no tryptophan synthesizing enzymes in E. coli, no tryptophan synthesizing enzymes in E. coli. Suppose we will explain it like this. We have this is a culture medium, this is culture medium. Suppose this is a petri dish, this is petri dish, this is petri dish, this is culture medium, and these are E. coli, these are E. coli. So we have got a petri dish, culture medium, E. coli, and tryptophan. This is tryptophan molecules. So when the tryptophan is added in the culture medium, we find no enzyme synthesis occurs in the E. coli. No tryptophan enzyme synthesis occurs in the E. coli. So no tryptophan synthesizing enzymes in E. coli. This is the case second. But what will happen next? In contrast to this, if here tryptophan is present, now if the case first, for example, we have same, this is petri dish, this is culture medium, and this is E. coli. Now here tryptophan is absent. When the tryptophan is absent, enzyme synthesis occurs in E. coli. So we have two cases. Case first is that when the tryptophan is present in the external medium, when the tryptophan is present in the culture medium, we find that there is no enzyme synthesis, no tryptophan enzyme synthesis occurs in the E. coli. And when tryptophan is absent, that means enzyme synthesis occurs in E. coli. This is contrast to the lac operon, because in case of lac operon, when the lactose was present, enzyme synthesis occurs. So that is an inducible operon. But here, this is a tryptophan operon is a repressible operon. That means when the tryptophan is present, no enzyme synthesis occurs. When the tryptophan is present, no enzyme synthesis occurs in E. coli. So lac operon is an inducible operon. Tryptophan operon is a repressible operon. Now, how will I explain this mechanism? What is the molecular mechanism behind this? When the tryptophan is present, no enzyme synthesis. When the tryptophan is present, no enzyme synthesis in E. coli. When the tryptophan is absent, enzyme synthesis occurs in E. coli. How we can explain it at the molecular level? This tryptophan operon, what is a repressible operon? It mostly operates in anabolic pathways. What we call it biosynthetic pathway. It operates in anabolic pathways. anabolic pathways, that is biosynthetic pathways. What are called as biosynthetic pathways. Because of the fact that in this case, in tryptophan operon, tryptophan is synthesized. That means this operon operates in biosynthetic pathways. Whereas in case of lycoperon is an operating in catabolic pathways, because in lycoperon lactose is being degraded into glucose and galactose. Here in case of tryptophan operon, which is a repressible operon, here tryptophan is being synthesized by tryptophan synthesizing enzymes. So we can explain it like this. We have the tryptophan operon components. Tryptophan operon components. What are the tryptophan operon components? What are the tryptophan components? We have same SOP. We have structural genes, we have operator gene, we have promoter gene. So we would have like this. This is structural genes: tryptophan A, tryptophan B, tryptophan C, tryptophan D, tryptophan E. These are structural genes. What are called as cisterns? Cisterns or structural genes. Then there is an operator gene. S means structural genes or cisterns, O operator, and P is promoter. This is operator gene. Then we have P, promoter gene. Then another gene that is called as R gene, regulator gene, and this is the promoter of regulator gene. So first we can explain them one by one. This structural genes, what are called as cisterns. What are called as cisterns. So we have tryptophan A, tryptophan B, tryptophan C, tryptophan D, tryptophan E. These tryptophan A, B, C, D and E, they synthesize what? They synthesize tryptophan synthesizing enzymes. They synthesize what? They all synthesize tryptophan synthesizing enzymes. Synthetase enzymes. They synthesize tryptophan synthetase enzymes. Then we have got O gene. This is O, operator gene. This is operator gene, what is called as O gene. This controls the expression of cisterns or structural genes. It controls the expression of structural genes. Expression of structural genes. Then we have got P, promoter. Promoter, it is a binding site for RNA polymerase. That means RNA polymerase binds to the DNA at a specific site that is called as promoter. That binding site for RNA polymerase. Binding site for RNA polymerase. Then we have got R gene. This R gene, it synthesizes protein that is called as repressor protein. Here it is called as, this is repressor protein. But here repressor is inactive, that is called inactive repressor protein. What is called as aporepressor. Aporepressor. Regulator gene synthesizes an inactive repressor protein, what is called as aporepressor. So this was about the, so we have, this is operon, this is tryptophan operon. This is tryptophan, operon. What is operon? Operon, it is the functional unit of protein synthesis. It is the DNA segment which controls protein synthesis. So it is composed of SOP, structural gene, operator gene, and promoter gene. So in case of tryptophan, operon, we have got many structural genes or cistrons. We have got tryptophan A, tryptophan B, C, D, and E. A, B, C, D, and E. And these synthesize tryptophan synthesizing enzymes. This O gene, what is called as operator gene, it controls the activity of cistrons or structural genes. Then we have got P gene, promoter gene, it is the binding site for RNA polymerase. Then repressor regulator gene, R gene, it synthesizes a protein what is called as inactive repressor protein, and here it is called as aporepressor. And this PR, it is the promoter for the regulator gene. So we can say that here this is the binding site for RNA polymerase. Same is the case, this is the binding site for RNA polymerase. Binding site for RNA polymerase. So what is the mechanism behind this? We'll discuss the molecular mechanism in case of tryptophan, operon. So first we will take case first. Case first means when external tryptophan absent, enzyme synthesis in E. coli. That means tryptophan absent, enzyme synthesis in E. coli. Enzyme synthesis in E. coli. This is case first. We will discuss this case first here at the molecular level. So we know that this is E. coli membrane. This is E. coli membrane. Here external tryptophan absent. That means tryptophan is absent here. No tryptophan. No tryptophan in the culture medium. No tryptophan. No tryptophan. Here what will happen? See the molecular mechanism, how enzymes are synthesized in E. coli. This is the bacterial DNA. This is like A. This is tryptophan A, tryptophan B, C, D, and E. 1, 2, 3, 4, 5. This is bacterial DNA. Yeah, E. coli DNA we can say. Then we have got operator gene, promoter gene, regulator gene, and this is promoter for regulator gene. So this R gene, this is the promoter of regulator gene. It will be a binding site. Promoter is the binding site for RNA polymerase. So this is the binding site for RNA polymerase. The binding site for RNA polymerase. This regulator gene, it will undergo transcription. It will synthesize repressor mRNA. This is transcription. Obviously, it will undergo translation. What will form? It will form an inactive repressor protein. This is inactive repressor protein. This is inactive repressor protein. Why it is called inactive repressor protein? Because this repressor protein is not able to bind. This inactive repressor protein is not able to bind to the operator gene. So it is called as inactive repressor protein. Here it also called as aporepressor. It is called as aporepressor. This inactive repressor protein, it is not able to bind to the operator gene of the operon. This is operon, tryptophan operon. So this is P, it is the binding site for RNA polymerase. It is the binding site for RNA polymerase. Now this RNA polymerase can easily go ahead and cause the transcription of the structural genes of the tryptophan operon. That means this RNA polymerase, it can move easily and cause the transcription of structural genes or cisterns of the tryptophan operon, because of the fact that the inactive repressor protein had not bound to the operator gene. That means operator gene is open. The road for the RNA polymerase is open, so it can easily transcribe the structural genes. So obviously here would be transcription. It will form what? Polycistronic mRNA. Polycistronic mRNA, it is characteristic feature in case of prokaryotes, where we find mRNA, it is having the information. Polycistronic mRNA is that type of mRNA which has information for the synthesis of many proteins, which can act as enzymes. So it can synthesize many proteins. So we have this. This is translation of polycistronic mRNA. It will synthesize. This is proteins which will act as enzymes. These are proteins. So these are proteins which act as enzymes. These are called as tryptophan synthetase enzymes. These are tryptophan synthetase enzymes. Tryptophan synthetase. These are tryptophan synthetase enzymes. And what they will do? They will convert the raw material, chorismic acid, into tryptophan. So these tryptophan synthetase enzymes, they will convert the raw material in the form of chorismic acid into tryptophan. Tryptophan is an amino acid. Tryptophan is an amino acid. Tryptophan is an amino acid which is required by the E. coli. So when it is as simple as this, when there is no tryptophan in the external medium, the E. coli has the capacity to synthesize the tryptophan. How it synthesizes the tryptophan? First, it synthesizes the tryptophan synthetase enzymes. Those tryptophan synthetase enzymes synthesize tryptophan amino acid from the raw material, chorismic acid. That means there is an inbuilt information in the E. coli, in the DNA, to synthesize the tryptophan synthetase enzymes, which are responsible for synthesizing the tryptophan in E. coli. This is the condition when the tryptophan is absent in the culture medium. But when you add extra tryptophan in the culture medium, then this tryptophan operon will turn off. This was when there is no tryptophan in the external medium. When no tryptophan is present in the external medium, the E. coli has capacity to synthesize the tryptophan amino acid. How it synthesizes tryptophan? It is operon. It is tryptophan operon remains on. That means it is tryptophan structural genes remain on. They are expressed, and ultimately they synthesize tryptophan from the raw material. This is the mechanism for the tryptophan synthesis. Now there is another case. In the case where you will add, this was no tryptophan. Now in the another case, we add the tryptophan in the culture medium to see how this E. coli will turn off its structural genes, because you have added the tryptophan from the external medium. There is no need for the E. coli to synthesize the tryptophan when it is getting a ready-made tryptophan. So there is no need for the E. coli to synthesize the tryptophan, as it is getting the ready-made, it is getting the external tryptophan. So now we'll just Now this is a case first. We'll discuss the case second. That is when tryptophan is present in the culture medium. Suppose we have this case second, that is tryptophan present in culture medium. So you have got this E. coli membrane. This is E. coli membrane. These are this tryptophan present in external medium, tryptophan present in culture medium, tryptophan present in culture medium, no enzyme synthesis, no enzyme synthesis, no tryptophan enzyme synthesis, no enzyme synthesis. Suppose this is tryptophan molecules, these are tryptophan molecules, in the external medium. We have the E. coli DNA. We have tryptophan A, B, C, D and E. This is E. coli DNA. And we have tryptophan A, B, C, D and E. Then SOP, these are structural genes, yeah, cistrons. Then operator gene, promoter gene, regulator gene, and this is the promoter for regulator gene. So this is operon, that is what is called as tryptophan operon. Operon is the structural and functional unit of protein synthesis. Operon is a segment of DNA which controls protein synthesis. So obviously now these, as this regulator protein, it will synthesize what? Repressor mRNA. This is repressor mRNA by means of transcription. Then translation will take place. So we have got what? This is inactive repressor protein. This is inactive repressor protein. What is called as aporepressor. Inactive repressor protein. Now you have added this, what? You have added tryptophan molecules. These are tryptophan molecules. Tryptophan molecules have been added in the external medium. Tryptophan molecules. These tryptophan molecules, this inactive repressor protein, it will form a complex. With tryptophan molecules. So it would be like this. This is inactive repressor protein. And this is what is tryptophan molecule. So it is a complex. Complex of what? Complex of inactive repressor protein, what is called as aporepressor plus tryptophan. This is tryptophan, and tryptophan here is called as co-repressor. The tryptophan here is called as tryptophan molecule is called as co-repressor. So this aporepressor, that is inactive repressor protein, this is this, and tryptophan molecules, what are called as co-repressor, this is this. It will form a complex. So it is co-repressor. What is called as aporepressor co-repressor complex. And this aporepressor co-repressor complex, this becomes active repressor. It is called as active repressor now. this aporepressor co-repressor complex together, it forms the active repressor. This is active repressor. Now this active repressor has the capacity to bind with the operator gene. That means it can bind with the operator gene. It can bind with the operator gene. This complex, which is formed of aporepressor and co-repressor. Co-repressor is tryptophan molecules and aporepressor is simply inactive repressor protein synthesized by the regulator gene. When these two combine, they have got an affinity with each other, and ultimately they form a complex together, and this complex is called as active repressor. This active repressor can now bind with the operator gene. So it has bound to the operator gene. When it will bind to the operator gene, so active repressor binds to operator gene, and it blocks the road for the RNA polymerase, and blocks road for RNA polymerase, and it blocks the road for RNA polymerase. This is a promoter where RNA polymerase is present. This is the RNA polymerase. Also, this is promoter RNA polymerase. This is promoter RNA polymerase. It blocks the road for this RNA polymerase. This is RNA polymerase. This is RNA polymerase. Now RNA polymerase, it cannot go ahead to transcribe the structural genes of tryptophan operon because the road is blocked at the operator site, and this road is blocked by the active repressor, which is composed of aporepressor co-repressor complex. So when RNA polymerase cannot go ahead, it cannot move ahead to transcribe the structural genes of the tryptophan operon. That means structural genes switch off. Structural genes are switch off. No transcription, no translation, no enzyme synthesis. So we can say that this is a simple mechanism, that when you have added the tryptophan molecules in the external medium, that means this information will go into the DNA of this E. coli, that it will not synthesize the tryptophan synthesizing enzymes, because when you provide the ready-made tryptophan to the E. coli, there is no need for the E. coli to synthesize the enzymes which are needed for the synthesis of tryptophan. So it means that at that time, when there is external tryptophan present, this operon is switched off. It is switched off. No transcription, no translation. So this is called as repressible operon. That means when the substrate is added to the culture medium in the form of tryptophan, no enzyme synthesis occurs in the E. coli. So this is called as repressible operon. This repressible operon, it is also a negatively controlled operon. It is also negatively controlled operon. This is negatively controlled operon. Why tryptophan operon is a negatively controlled operon? Because of the fact that this repressor protein, inactive repressor protein, it combines with the co-repressor and ultimately inhibits the transcription of the structural genes. So it is a negatively controlled operon. Negatively controlled operon is that type of operon where the product of the regulator gene, will inhibit the transcription of the structural genes. Here the product of regulator gene, that is aporepressor, in collaboration with the co-repressor, that is tryptophan, they have inhibited the transcription of the structural gene. So it is a negatively controlled operon. Now for example, if there is an optimum optimal level of so this was about the mechanism of the tryptophan. If there are, for example, when we add the extra tryptophan, when we add the when we add the tryptophan in the culture medium, no enzyme synthesis occurs in the E. coli. This E. coli has, for example, E. coli, it requires optimum level. It requires this E. coli. It requires, it needs 10 molecules of tryptophan. It requires 10 molecules of tryptophan. Now this E. coli, it will synthesize the enzymes which will synthesize these 10 molecules of tryptophan. Now if we add externally this tryptophan, that means if the level of tryptophan will go beyond the 10 molecules, the concentration of tryptophan molecules in the E. coli is more than 10, then this tryptophan operon will switch off, because there is no need for the E. coli to synthesize the tryptophan when it is present in the external medium, when it is present in the optimum level in the external medium, or when it is present in the optimum level in the E. coli. So we can say that, for example, this E. coli turns off tryptophan operon. E. coli turns off tryptophan operon if the tryptophan molecules in E. coli are present more than what is required. That means E. coli, it turns off tryptophan operon if the tryptophan molecules in an E. coli are present more than what is required by the E. coli. It means that, for example, if E. coli requires 10 tryptophan molecules, it is requirement. If E. coli requires 10 tryptophan molecules, it is requirement. And there are more than 10 tryptophan molecules in E. coli, then operon off, no enzyme synthesis. So why it is? It is because of the fact that so as to conserve the energy, because formation of enzymes for transcription, translation, it requires an expenditure of the energy. So in order to save the energy, in order to prevent the loss of the energy, in order to conserve the energy, what it does, this E. coli, it stops the operon when there is more than optimum level, more than needed level, a required level of tryptophan molecules in the E. coli. So this is basically the basic underlying principle in case of the tryptophan operon. This tryptophan operon, it can be explained by means of a graph, that how the tryptophan addition decreases the this statement I will explain with the help of a graph. For example, we have got this, we have x-axis, we have y-axis, horizontal is x-axis. We'll take a time period in the form of minutes. We have got 0, 5, 10, 15, 20, 25, 30, and here we have got what? Enzyme activity. Here we have got enzyme activity. Here we have got enzyme activity in tryptophan biosynthesis. This is enzyme activity during tryptophan biosynthesis. This is the time period. So when we add the tryptophan, enzyme activity is very high when there is no tryptophan, but when we add the tryptophan, the enzyme activity in an E. coli decreases. It will be like this. It will decrease. So it would be like this. You have added what? You have added tryptophan. Tryptophan added. Tryptophan added. When you have added the tryptophan, that means the enzyme activity. That means the tryptophan enzyme synthetases. Their activity will decrease. That has decreased with the passage of time, and ultimately it becomes constant. So it means that addition of tryptophan has this is a repression in enzyme synthesis by the addition of substrate, by addition of tryptophan. That means when tryptophan is added, the enzyme activity in E. coli, the activity of enzymes which are involved in tryptophan synthesis, that have decreased with the passage of the time, and ultimately it has become a constant. So we can say that there is when we have added the tryptophan, when you have added the tryptophan in the external medium, we find there is a repression, that is a decrease in the activity of the enzyme. So this is repression. So this was regarding the tryptophan operon. Then how this molecular mechanism which is involved in the tryptophan operon. Thanks.